back to the structure of aorta. As you can see on the right hand side, you have the structure of the artery wall, in which we have three layers. One is the tunica externa, which is the outermost layer of the aorta. We have tunica media, which is comprising of the muscle tissue layers, and then we have tunica intima. Now, most of the nano indentation tests which we performed were on tunica media, because tunica media is comprising of muscle cells which actually have the mechanical properties changing across the X, Y, and Z direction, which also implies that they have anisotropy. The aims and objective of this research were to measure the viscoelastic properties, because we needed to understand that how exactly the mechanical properties are changing in the frequency domain, and how exactly the stiffness values changes as we go from the macro length scale to the micro length scale. It will also enable us to better understand the aortic degradation, because ultimately the mechanical properties changes in case of aortic degradation. Most importantly, we were able to fit some of these mathematical models, which will also enable us to extrapolate the models from less than 10 hertz of frequency up to frequencies in the orders of megahertz, which actually is the case in case of dynamic microelastography. So these models can also act as something which is validating some of the results obtained from dynamic microelastography. Then we were using these fractional order derivative models, because compared to traditional viscoelastic models where we need a large number of parameters, we can actually use spring bots, which are like fractional order elements, where we can characterize the properties of aorta in the frequency and time domain by using fewer parameters. In terms of application, one is that these models can be embedded into the finite element analysis, because in case of, for example, say pulsatile blood flow, and this is one of the, I would say, images obtained from this particular paper, where you can see the wave speed, and you can also see the wall stress changing as we have the pulsatility in the blood flow. So most of these models can be embedded in case of the finite element analysis softwares like Abacus or ANSYS, and then we can actually use them to, because these are computationally less expensive, so they can be utilized well in a short period of time. So there were three types of tests. One is the strain sweep test, which we conducted, and the other one was a frequency sweep test, and then in collaboration with Dr. Riaz Akhtar, it was the narrow indentation test. So you can see some of the strain sweep test here. On the right hand side, you can see the this particular aorta that has been obtained from a pig, and you can see some of the lungs there, thoracic region, which is consisting of the proximal descending, mid descending, and the distal descending region, and then the abdominal region of the aorta as well. On the left hand side, you can see this particular setup which I was using. That's a setup of Physica MCR 302, which is a stress controlled rheometer. Now, how this particular setup is working is that you have a stress controlled oscillation in which you apply a torsion, some sort of a torque to the cylindrical samples of the aorta. And as a result, the cylindrical samples are applying a reactionary torque. And that particular reactionary torque is nothing but the storage modulus. That is the amount of energy that is being stored in the sample. The amount of energy that is being dissipated is also measured as something known as the loss modulus. So we had 1.5 mm thick aortic samples. I was actually able to cut them into very nice 8 mm diameter samples, which you can see there. And the locations actually vary from distal descending region, proximal descending, mid descending, ascending, and the abdominal region. And all these samples were immersed in the PBS solution, because we also needed to make sure that we are able to hydrate these particular samples across the testing. So this was one of the key takeaways. So one is that we were able to see some of, I would say, linear viscoelastic region actually in all the samples, and most importantly, in all of the samples, the linear viscoelastic region was until slightly greater than 0.1% strain, which also implies that at this particular value of the strain, both the storage modulus and the loss modulus are pretty much intact. And after this, we either have a nonlinearity or we have a damage caused in the samples. So this also told us one interesting thing, because once we were able to understand the threshold limit of the shear strain, we also knew that at what value of the shear strain can we actually perform the frequency sweep test, because I think that's the most important part, because we were actually fitting some of these models there. So in case of frequency sweep test, again we had 5 porcine aortas tested within 12 hours of slaughter. Again, the locations were exactly the same. In terms of frequency, we were able to perform them until 5 hertz. The reason we actually only did until 5 hertz is that because above the frequency of 5 hertz, we had the inertia. And in case of inertia, what happens is that there is a slippage between the top plate of the rheometer and between the aortic samples. So on the right hand side, you can see this particular equation number 1. So equation number 1 is the equation of the stress strain of a here, if you actually substitute the value of alpha equals to 0 in this particular equation, the spring port is changing to that of the spring. At the value of alpha equals to 1, we have the spring port changing to that of the dashpot, which implies that this particular element is actually having properties which is intermediate between that of the spring and the dashpot. So once we convert this particular equation from time domain to frequency domain, we can actually obtain one set of equation for the storage modulus, which is equation number 4, and the other set of equation for the loss modulus, which is equation number 5. So I was fitting these particular equations using the software like MATLAB, performing some of the mathematical modeling there, and then extracting these particular parameters, which is k alpha and alpha. Now, k alpha is the coefficient of consistence, which is nothing but it is taking into account the rate of binding and the unbinding of the, I would say, collagen, elastin, and the interstitium. Because across, as the frequency is being applied, there is some sort of binding as well as the unbinding, which has been quantified using this particular parameter known as k alpha. Alpha is the order of fraction, which implies that it tells us that how exactly these particular samples are dependent upon the frequency. So which implies that the higher the value of the alpha, so for example, say in the ascending region, it also implies that the storage modulus is increasing drastically as a function of frequency. In case of the ascending region, it is actually increasing less, for example, say in case of proximal descending and the distal descending region. So we were able to quantify these particular parameters across different regions of the aorta. So you can see this particular figure here. So this is the storage modulus as a function of frequency from 0.1 until 5 Hz. And also the loss modulus. So you can see like a very smooth behavior of Aadarsh across different regions of the aorta. And this particular model was able to fit like all the regions of aorta with a correlation which was higher than 0.99. So that's something which is good. So when I tried fitting some of the basic models like SLS model, standard linear solid model, or Maxwell model, or the Kelvin point model, it was not able to take into account this particular behavior as a function of frequency. There are some of the models, for example, say Prony series, where you actually apply like a series of springs and dashboards along with the Maxwell model. But the problem is that the computational expense actually goes up. So that was basically one set of the experiments. The other set of experiments was using a nano indentation setup in which we had a coil magnet assembly. So you can actually see the schematic of the G200 setup, which was utilized here. On the left hand side, you can see the coil magnet assembly, leaf spring, capacitive gauges, and the indenter. So essentially, we were trying to increase the frequency of the indentation from 10 Hz. The reason we actually went down to 10 Hz is that because we were also trying to understand if at this particular frequency we were able to see a fair enough comparison between the micro length scale and the macro length scale. Because at the macro length scale, we went up to a frequency of 5 Hz. So on the right hand side, you can see the G200 setup here. So this was the materials and methods in which we had the dissected aorta, which you can see here. We utilized a 100 micron indented tip. The reason being because we were actually wanting to, so I mean there were two things to it. One is that we were actually wanting to test at the micron length scale, that was one aspect. The other one was that we tried to use a flat punch because compared to other punches, the creep, so basically in case of viscoelastic tissues, we also have the property of creep, where the strain and the interaction between the tissue and the tip of the indenter actually changes with time. So to avoid that and to make sure that we had a surface area which is constant across these particular indentation, we were using a flat punch tip. All the tests were conducted like at 10 Hz, and also it was gradually increased up to 110 Hz, which is the resonant frequency of this particular actuator. The oscillation amplitude was around 500 nanometers, which is 0.5 microns, and the pre-compression was around 5 microns. So the process was we were able to obtain some of these rectangular samples, place them in the rigs, select the indent points. In this case, it was 3 indent points, and then perform the process of nano indentation. So the other interesting observation was that as we were going from the ascending region of the aorta to the abdominal region of the aorta, we were actually able to see a very smooth increase in the storage modulus. So the stiffness in case of the storage modulus actually increased from 10 kilopascals, which was around the ascending region, to approximately 55 kilopascals, which was in the abdominal region. In terms of loss modulus, I was not able to get like the same relation between the stiffness versus the region. I mean, there might be different reasons to it. One of it can be that in case of the storage modulus, it's basically nothing but the elastins and the collagens which are playing a role in the stiffness compared to say the interstitium, which is not exactly replicating well across these particular regions. And then when the indentation frequency was increased up to 100 Hz, we were also able to obtain an increase in the storage modulus across these particular regions as well. So as you can see here that at the ascending region, the storage modulus was approximately 10 kilopascals, but as we are increasing the frequency, the storage modulus is also increasing across all these particular regions. So it goes from approximately 40-45 kilopascals to around 100 kilopascals. Again, the same effect was not observed in case of loss modulus, and I'm still trying to understand why. But the potential reason can be that the interstitium is not well replicating this particular behavior, what we are seeing in case of the elastins and the collagens. So in conclusion, one of them was that we were actually able to quantify the properties both at the micro length scale and the macro length scale. I was actually able to extract this particular linear viscoelastic region until 0.1% shear strain. There was a monotonic increase in the storage modulus and loss modulus as a function of frequency, which implies that there was a power law behavior across different regions of the aorta. The spring-Mott model also exhibited the best fit for the frequency response, and compared to, for example, say Maxwell model, Kelvin-Voigt model, traditional viscoelastic models, and even the SLS model, it was actually having a better correlation coefficient, which is good. The other thing which is good about it is, like I was mentioning, that with fewer number of parameters, we can actually quantify the behavior of the aorta in the frequency domain. Then the interesting part was that in case of the storage modulus and loss modulus at the macro length scale, we observed a behavior which was different in terms of the dependency of the frequency, as in some regions actually were more dependent on the frequency compared to, for example, say the other regions which were slightly less dependent on the frequency. So it actually went from 0.1 to 0.6. And then the macro shear storage modulus increases from the ascending region to the abdominal region, and the same is also valid in case of the micro scale storage modulus. One of the other, I would say, interesting part was that the micro length scale storage and loss modulus, specifically the storage modulus was approximately 10 to 16 times higher as compared to the macro length scale storage modulus. The potential reason being because I think these particular collagen and these elastins, they're actually behaving far more stiffer at the micron length scale compared to the overall bulk tissue, because in case of the overall bulk tissue, we actually had like a huge, I would say it's like n number of layers of elastins and collagen one above another. Each and every layer is having its own relaxation modulus. Each and every layer is also having its own elastic modulus. So I think it's basically the combined effect of both elastins, collagens, relaxation, and also the interstitium, which is playing a role as far as the macro length scale is concerned. So in terms of acknowledgements, a big thanks to the NIH because they were funding our research, Dr. Riaz Akhtar because he's the main supervisor of this particular project collaboration, which is between University of Liverpool and Oxford. Dr. Martin Hozak, I think he finished his PhD recently in Department of Mechanical Materials and Aerospace Engineering, and my supervisor, Professor Robin Cleveland, because we went to this particular Biomed H21 conference where I was able to collaborate with Dr. Riaz Akhtar. So that's it, and thanks to all of you for listening to this particular research and giving me this particular